right. Well, good afternoon, and welcome to our fourth and final webinar as part of the OMOP webinar series, From Research to Regulators, effectively using SDTM and OMOP. As always, I'm very glad to be a part of this webinar series here, moderating, and thank you all once again for your enthusiastic participation across through all of our past webinars as well, and as to this one. It's because of your enthusiasm and eagerness to participate is that we also find ourselves very eager to be a part of to keep hosting our webinar series and keep it going. Well, if you have been have been following our webinar series, that you know that during the last time we had our wonder Guus Wilmink from the Hive and Theo Stocker from InterSystems, and we talked about two key topics, namely FHIR and OMOP, and how they can work together to advance data integration and research in healthcare. And today we are going to dive a little bit more deeper into two standards, namely the OMOP CDM and the SDTM, which is basically our clinical trial data standards. Julia and Berber here are going to be telling us about the striking contrasts of it, the similarities of these systems, and how they compare with each other, and how we can, in fact, use them effectively. And if you are joining these webinar series for the first time, please don't worry. Julia will make a wonderful introduction, as well as Berber, about these topics before diving them deep into it. And on that note, my name is Archita Keizers. I am a biomedical research business analyst at the Hive. And I'd like to give the opportunity now for our speakers to introduce themselves. Berber.
Thank you, and also thank you for inviting me here to co-present at this webinar. So my name is Berber Snoeier. I'm also a digital data flow consultant. I have more than 25 years experience in the industry, of which I also worked 8 years with real-world data as an R&D manager. So looking at what can we do with the source, the data that we get from electronic health records. In 2018, I founded ClinLine. And we focus on the optimization of the clinical data trial process, and also how we can utilize real-world data in that process. And with that, we look at different implementations. So ClinLine is dedicated to optimizing the efficiency in clinical data flow for regulatory submissions. And with that, we combine technical skill sets with content knowledge and business process insights and experience. And I am also involved in industry-wide processes like the Transcelerate CDISC Digital Data Flow Initiative, and I'm chairing the FUSE Real World Evidence Working Group.
Yes,
all right. Thank you for that.
Then I will take over here. So my name is Julia. I'm also happy to co-present with Berber today and to also be part of this webinar series. And so my name is Julia. I have a background in chemistry and neurosciences, and I work at the Hive for almost 10 years now in different roles, from business analysis, project management, and now more the real-world data consulting. And so I'm also involved in a lot of real-world data projects since, actually the last 8 years, and actually helping a lot of industry parties, but also academic parties, research institutions to adopt the OMOP CDM and the tooling around that. And I'm also involved in a lot of public-private partnerships where industry and academia actually work together. And so I'm also going to present some of the big projects that we work on. One of them is Eden, and the other one is the Darwin EU initiative. And I'm really looking forward to the conversations and to your questions today. And I would also like to shortly introduce the Hive. So if you have seen some of the other webinars, then you probably know already what we do. But I'm just going to repeat it again. So what we're doing is we focus mainly on open science, and we do support that via 3 different pillars. So we work with a lot of open source solutions where we either develop new features, for example, or we help our customers and partners to implement those toolings. The second pillar is verification of data. So assuming that you're a data holder, you have some data and you want to make sure it's more fair, so more findable, accessible, interoperable, and reusable. So it can be used for secondary use and in collaboration with other parties. And then we help our partners and customers to find out what are the right standards to use and how can you create semantic knowledge graphs, for example, and models. And the last pillar is the expert consulting, since we have quite a broad range of expertise here in the company, from cancer genomics over drug discovery to real-world data. And so we're located in the Netherlands, and we're founded in 2012. And yeah, I think it's important to say that we have a lot of expertise on a lot of different life science domains. And so since we're going to focus on two different standards today, I would like to introduce the OMOP CDM as a starting point here. So I think also this, if you have been to the other webinars, you have probably seen that before. But I think it's a good reminder. In 2007, there was a public-private partnership that focused on drug safety surveillance, and this was called the Observational Medical Outcomes Partnership. And one of the deliverables, or some of the results of this partnership, was actually the creation of a common data model, which was the first version of the OMOP CDM. And so then in the coming years, there were a lot of different versions of it. And in 2014, the OHDSI community was founded around this data model. And OHDSI stands for Observational Health Data Science and Informatics. And so it's not only the data model, but it's also tooling to convert the data to this standard, but also to analyze the data. And so in 2014, we had the fifth version of the CDM, which also evolved over time, of course. In 2018, there was version 6, which has been developed, but is not really integrated in all of the tooling, so that's not used by the community really widely. But version 5.4 is the one that's stable since 2020, and it's being used by the global community worldwide. Okay, and then I say, of course, the OHDSI community, because it's not only the data model, but it's also a global community of thousands of researchers that work together. They develop the tooling together, and there's also a lot of resources that you can find online, for example. So there's different courses, there's a lot of YouTube videos, and there's also a lot of publications coming out of this community every year. And if you're interested in learning more and joining, then I would definitely encourage you to go to one of those face-to-face events, because they're quite a lot, and they are also scattered around the whole globe. I think it all started out with the Global OHDSI Symposium, which is always in October in New Brunswick, New Jersey. And then there's a European one and the Asian Pacific one. The next European one will be in April 2026 in Rotterdam, so just around the corner from us. All invited, of course. And then there's also other symposia in Africa and India, which are really recent, but you can see there's a truly global community that's really growing. But coming back to the model, this is a representation of the model. It's a person-centric model, and it has 37 tables. It's mostly used for observational research. So the fields that are most important are, of course, the person table for the demographics, but there's also condition occurrence, where, for example, all the diagnoses and all the diseases a person have are stored. Then you have the drug exposure table, where all the medications, for example, are stored. And you have procedure occurrence, which is, for example, surgery or other therapies. And so what you see is that there's, beside the standardized clinical data, you also have some tables that focus more on health economics. And the other important point for the CDM is that it's not only the model, but what you see here in the middle are standardized vocabularies, which are also maintained by the community. There's regular updates every 6 months. And this is really something that helps the community assign the data and the concepts that are standardized. And I think this is the basis, what you need to know for the OMOP CDM. And then I think I would like to give the floor to Berber to tell you a little bit about SDTM.
Yes, maybe in the meantime that we have this transition, we can also have a little poll question that we have here to try to understand the type of data that our audience here works with the most. Is it a lot of clinical trial data, or is it a lot of real world data, or is it equal? Or are you neither and it's quite new for you, these topics, and you're just here to kind of figure your ground out? So we would be eager to know about where you stand here. So please feel free to enter your answers here. Alrighty, we already have pouring votes, and I think more than 50% of them come a lot for real world data. So in terms of EHR claims and registries data, so that looks good. But also, then at a close equal is then either both of them or neither of them. So yeah, I think we have a strong preference towards on one hand. So that's good to see already the real world data. No.
Okay, I hope you can hear me now. So yeah, after the introduction of Julia, I can now tell a little bit more about CDISC and their SDTM data model. So that started in the start of this century, or around the start of the century. CDISC started with the SDTM data model, and it got incorporated into the standards of the pharmaceutical industry when also this standard, this data was required, our clinical data was required to be submitted in SDTM format. So this is actually the regulatory submission standards.
You're not, sorry to interrupt, but you're not sharing your screen.
Oh, that is, I need to do, yeah, there's a bit of a, let me try again.
Yes.
Apologies for that.
But to our audience who's already here, if you are just joining right now, then fret not, because we will have a recording of this session that will be available, and the slides as well that will be shared to you. And of course, if you have any questions, please feel free to drop them in the chat, and Julia and Barbara will answer them for you as well.
Can you see it now?
And now we can see your slides. Yeah.
Perfect. So I was talking about that CDISC SDTM is actually the regulatory submission standard, at least for the FDA, but also in Europe, it's starting to become a standard that is required for submitting clinical research data. And actually, what Julia showed about the OMOP data model, this is combining the clinical data part and the study design. So, for example, the terminology is not stored in tables. It's defined in their terminology lists that are also defined by NCI. The CDISC SDTM data model has a great variety of domains and is also aligned with other data models within CDISC, for example, for the analysis item, for the collection, etc. So there are different data models, but the SDTM is really the basic clinical data. So like for each domain, it is defined what are the variables, what are the labels, and also what control terminology lists need to be used for that. And also what is good to note is that in 2024, early 2024, they added a small implementation guide around observational studies and real world data, which is helpful, but I think for the future, we are also looking into how can we further guide the industry on that. So yes, there is a lot of new guidelines from regulatory perspectives issued in the last few years. And to start with that, the definition is very important for real world data. It is, according to the FDA, data relating to health, to patient health status and or the delivery of healthcare routinely collected from a variety of sources. And that routinely, that is the most important part here, because that is like, okay, it is not collected for the purpose of the trial. It is really for treating the patient as good as possible. So that information that is needed for that is collected well, but other information that you might need for clinical trials, maybe not. EMA has a comparable description, data that describes patient characteristics in routine clinical practice, and also the PMDA has a related description. So if we compare clinical trials with real-world data studies, then we see a lot of differences. Clinical trials are usually randomized, while real-world data studies are not. Clinical trials have that data collected for the purpose of the trial. You can really dedicate what exactly is collected. And while for real-world data, it's coming from those real-world data sources that are collected in routine practice. Clinical trials, data largely collected via EDC systems, at least in the past, and real-world data sources. We collect the data from source information systems. Clinical trials, data processes according to decades of standard siloed processes and procedures. While for real-world data studies, we need more collaboration in our industry. We need to change processes in order to be able to use this data effectively. If we then look over time from one space to the other, then are also trials. It's not one or the other, but there is also some trials in between. So we have the randomized trial on the one side, which is completely coming from that EDC part. Well, we also have nowadays more pragmatic trials that are also including data directly from EHR sources. We have external control arm trials where we have one arm as an interventional arm, an interventional trial, and the other arm is coming from EHR data. And then we have studies that are completely based on EHR data, like the real-world data studies, for example, for new indications. And what I do mean with that is that if the drug is already approved for another indication, then it's already used in normal practice, but then we look into, okay, is it already used for new indications? What are the features of those patients and what are the effects for those patients? So when you look at that, data sources, EHR data sources, then you can have a lot of different data sources. It can be GP data, general practitioner data, you can have hospital specialist data, pharmacy data, laboratory, and different data sources. And each source has his or her own scope. So that collection practice for each source has an effect on the reliability and what you can analyze of that data, and what is collected has an effect on the relevance of the data. And those two terms are frequently also used in those regulatory guidances. So different, also within each of those data sources, you see a lot of different EHR information systems used. And that the system that is used results in variation within and between those domains. Maybe that they have different data structures, even within countries, between systems, within systems. You have different entry screens, etc. And all that has an effect on what does the data actually mean? How complete is it? What is really collected? And what is the interpretation of what is collected? So that is very important to keep in mind when you are selecting your data source. So if we look at the different types of data sources, then we see electronic health records and claims, and that is data primarily collected in the scope of treatment of patients in regular clinical practice. That can be GP or specialist data. We also have registry data, which is an organized system that collects clinical and other data in a standardized format for a population defined by a particular disease or a condition or a drug exposure. And then sometimes the data is directly entered into the registry, or it is combined with EHR data from other sources. And finally, the most difficult real-world data source that you may want to look into for trials is the patient-generated health data. And then you can look at social media data, also commercial device data, etc. So there's a lot of regulatory challenges when you want to use these data sources. And those guidelines and regulations, they want you to show the data lineage from the data source to your submission. So every step in that process needs to be shown and also needs to be governed. And so you need also to have often source access. So that means that you need to be able to go back to the source, and they need to be able to check, okay, this data that you have submitted for this indication, is that actually also, can I also find that back in the source? And that can be challenging, because if you anonymize your data, then that's of course not possible. Then, of course, assuring the reliability and the relevance of your data is also important. So when you select a data source, you need to look at all those different aspects of relevance and reliability. Do I have, for example, the data for the correct period? Many data sources have older data, like, okay, we have data from 2005 until 2015, but often that is not seen as relevant anymore by the regulatory agencies. So it must be more recent data. And there are many different relevant aspects, as you also can see in the EMA data framework. Also, what I already mentioned, that source collection variation, the heterogeneity in the information systems, the heterogeneity also in the treatment protocols and the practice, also how are patients treated in a certain area. There can be differences within treatment protocols between countries, but also even within countries. You have groups of specialists that have their own ways of treating their patients, but also their own ways of collecting that data. And then the extensiveness of the data that is collected, the representativeness and the missingness needs to be addressed. What is also noted in the guidelines is what standards are used. Those information systems, they use their own data model. And then when the data is exchanged, whether it's FHIR or whether it's OMOP or whether it's another standard, it is already mapped. There is already a step in between, and maybe some information could not be mapped well enough, so you're losing maybe information. So that's those transformations at the source that you need to be aware of. You need to be to understand that these things happen in the process. Finally, and nowadays we are using more and more AI with extracting data from text, etc. And that's very helpful, or maybe the AI for the mapping. But of course, these steps also need to be validated in order to be able to use that data for regulatory submission. So talk a little bit more about the data lineage, how it refers to the process of tracking how the data is generated, transformed, transmitted, and used across a system over time. And so that means that every step needs to be documented, from that electronic health data source to finally that submission in SDTM format. And so therefore it's important that you combine those different sources in one data standard, and then you can do your selection, etc. You can validate your data, and then you can select your data in your study cohort, and you can transform the data to SDTM. And why are we not using SDTM in an earlier stage? I will explain you in the next slide. But before I do that, I want to go a bit more into the lineage. So it's including the origins, the transformations, the movements, and that provides the detailed visibility in the life cycle. And then if you have a data point at the end, you know, okay, this is where it's coming from. This is where all the steps in between. So by that, you enable the users to trace issues back to their root causes. And that also, if I look at the regulators, they want to know, okay, if I have a data point that is not exactly as at the beginning, why is that the case? What happened? So that's also important. Includes all the procedures, contract, code, output, and documentation. So how do you standardize your real-world data? Now, there are different ways of doing that. I heard that the previous webinar was about FHIR. FHIR is a good exchange format. It's flexible, focuses on the clinical practice. It's a good exchange format, but it's not very handy in analyzing your data, doing fit for purpose as analysis. Doing it then directly to creating directly SDTM domains. As you saw already in my previous slides, there are many different domains, and also the coding is different than the source, so the transitions can be very cumbersome. Creating all those domains for data that you are not sure yet of that you want to submit to the regulatory agencies, that can be very difficult. So, and also not all the study design information is available in your real world data source. That is something you need to add at the moment that you have your study design specified. However, it aligns to interventional data, so that's helpful, and it's required for submission. OMOP then, it's that basic vertically aligned structure, as Julia has already mentioned, and it's already used for observational studies. So it's already looking at what is needed to do selection, select cohorts, etc. What I think is very helpful is that it includes the source variables. That's helpful for the traceability, the lineage as well. And it has extensive selection and mapping features. I also saw now that the SDTM, the CDISC terminology is now included. So transforming from CDISC to other formats is now also available via the OMOP mapping features. So the interoperability between the standards is, of course, challenging. You need to do some mapping, etc. So different data structures, different terminology. Sometimes you have missing information that you need. The governance of the different models is sometimes difficult, and also the usability. So therefore, from source to CDISC, it's sometimes difficult, also because you have different standards maybe at the source. Or you have FHIR, so then that in-between data layer is needed for fit for purpose. And I think that the OMOP or the OMOP standard is very helpful for that. Some companies do extended simplified SDTM, it's also an option, or others that use only one data vendor use the data vendor structures, which are often very much alike OMOP. Challenges. When we map OMOP to SDTM, then we did some use cases in this area, and then the challenges were that selecting the right data that you want to use for your study, but also the index data. If you define your study, you need to define an index data. One is the subject included in your trial, and you also need a different windowing than compared to your interventional arm, because you want to use as much data as you can from the real world data source. So you need to create rules for that, and those rules need to be governed. Also make a distinction between what are adverse events versus medical history. I don't have a lot of time to dive into that, but then the index date is very helpful, because everything before the index date would be medical history, and after would be then kind of an adverse event, maybe not related, but that's how we call it in the clinical trials. Same for concomitant medications versus previous and concomitant medication, but also the exposure if we are looking at a drug that is given in real world practice. What was also very challenging is what assessments could go to could you select from observations and measurements? I think this should be also in the second slide. And then the source to CDISC mapping. So I go from a Loing variable to CDISC, that's now available. However, the direct mapping can be quite difficult. Maybe the units is now also included. But what was also very challenging was the categorical results, mapping the categorical results. The other hand, SDTM to OMOP. That is the OMOP measurement and observation class. So if you have variables in SDTM, are they going, if they are not yet available in OMOP, are they then a measurement or are they an observation? Sometimes it's difficult to see that, to point to that. But also mapping of typical trial information to OMOP concepts like, is a drug ongoing or not, the arms, visit numbers and partial dates times. For that, you would need also rules. What What was also very challenging is the mapping of the events to OMOP or SNOMED. So not all the events in CDISC are actually condition occurrences. Sometimes it's just an observation. But when we did that, we got a 90% automatic mapping based on OMOP mapping capabilities. So that was very nice. The last thing was, again, that medication, which was very challenging to move the terminology and the standards from CDISC to SDTM to OMOP. So yeah, what is also a good point to note is the semantic interoperability. So going from the different coding systems, I already mentioned the medications and the indications, the events. But yeah, also other code lists like the route of administration was sometimes quite different. If you have different sources, like you see here in this slide, it could be that some values are available in one source and not in the other source. So this is important to know as a statistician that there can be a bias based on what was in the source. So that's why you need also that source information in your data sets. And then, of course, the mapping, do you have all the information available in the code list for each standard is also good to note. So that's really that governance and knowing what there is in. So you also, apart from the variable trailing, you also need to include the full coding trail from source to submission and address that misalignment that I showed here. We did a proof of concept also with the mapping of the patient data. So here we have the mapping via data, which is more that exchange format, put it in OMOP Common Data Model, and then I created SDTM data from that. We are still working on that, proof of concept, that use case, also within the CDISC real world data lineage project, where they are looking into, okay, how can we best submit that information then to the regulatory agencies with all that source information included. This is, I think I need to hurry up a little bit. So this is the data lineage use case that we are visualizing, and those visualizations really help to understand the process. So if you see long lines from source to a later step, then you might want to think, maybe we need to change that to make it more efficient, to make it more one line instead of all lines crossing each other. So yeah, you get insights in what is happening during the mapping, and afterwards, of course, if I have a variable here, then I can see how it goes back, and I can click on the transformation nodes, that's how we call these, to see what happens in each step. So those transformation nodes, that is really describing what happened between those two fixed stages. There can be the selection of the data, can be the validation of the data, can be the exchange of the data. And within each node, you describe the input, the output, all the mappings, the variable mappings, but also the code mappings, and link that to corresponding procedures, documentation, code maybe, etc. So here you see an example of the first level in the transformation node. So you have two different data sources which are linked. The linkage is also described in the background here. And then you have the input variables and all the actions that happen there. And it can be that it's just a copy, but it can be also a recode or a transformation, etc. And the idea is that you then click on this and then you see the details behind it. So yeah, summarizing this, what I did not yet mention is that in the regulatory guidances, and you see that from all the different regulatory agencies, it's very important to start with the research question of your clinical trial, not clinical trial, of your study, because otherwise you cannot select the best data source, because a data source that is good for one question might not be good for another question. And so then based on that, you can evaluate the potential sources and align with the regulators on that. Is this good data? Do you accept that? And after that, you can acquire the data. Make sure that you ensure the data lineage. And for that, it's better to define the requirements at the start. What you now see is that people are very much used to the standard processes. This is really new to use real-world data, and then, yeah, maybe the lineage comes later. Some documentation, there is documentation here and there, but afterwards you need to find it and put it in the right order. But that is, of course, less efficient than already at the start. Realizing all those needs for lineage and realizing that you need to be able to and have this available for the regulatory agencies. So for that, you can improve your processes by using visualizations, use tooling, maybe use AI, and then, of course, perform the validation. With that, I'd like to hand over to Julia again.
Thank you so much, Berber, for this talk. And as we expect, we have some questions as well in the chat. I think I would like to ask a question before Julia starts to share her screen. Ramiro asks a question about what software is used to tracking data lineage.
The software, the tool that I now showed is an open source tool that we created. So that is available on our CleanLine GitHub. It's still in development, but I think those first two steps are already available. But of course, your input needs to be aligned then. So, but you can find more information there.
All right, thank you. We hope that answers your question, Ramiro. And without any further ado, I'm going to hand it over to Julia.
Yep, perfect. Thank you. So I would like to use the remainder of the time to walk you through a couple of flagship projects that are really important when it comes to real-world data, OMOP, and regulatory use cases, I would say. And then I want to start with the Eden project, and then I'll tell you a little bit about Greg and about Darwin EU. We're starting with the Eden project. So it was an IMI project, so standing for European Health Data and Evidence Network. It started in 2018 and ended October last year. And the idea was here that this is a public-private partnership with people from academia, but also from the industry, working together on creating a different ecosystem when it comes to real-world data sources in Europe. So the mission was to actually build, what I said, a different ecosystem that allows us to find real-world data sources and to also do large-scale analysis over a network of high data quality resources in Europe using a common standard that would allow us to do this federated analysis. And so the vision, of course, is behind that is that we can use this data then to make better decisions for patients and for the outcomes. And so the values, of course, they align pretty much with what we also have here, so that you look at open science, that you use collaboration and work together with communities. And so I think what was really helpful in the Eden project is that we had two streams. So one of them is the data partners. So they are not called data holders or data providers because they're not only providing the data, but they're really partners in the collaboration. And so there was a harmonization fund as part of the project that also enabled those data partners, which could be hospitals, registries, so all the sources that Berber mentioned, those could use this harmonization fund to map the data to the common standard. In this case, it was the OMOP standard. And there was a different stream that you can see below, which is supporting SMEs. So SMEs is in small, medium enterprises all over Europe, which are trained and certified to do the OMOP mapping. So to help those data partners to get their data into the common data model. And so I think this approach really helped us to build that ecosystem, what we have now. On this screen, on this slide, you see the data partner journey. So what are the steps that data partner, for example, hospital, goes through in order to make their data ready for network studies, for example. So as you can see here, you start on the bottom left. Everything in green is concerning data, and everything in red is more about the infrastructure. And the little icons that you see here these are the tools that you can use that are developed by the Odyssey community, that are open source tools that you can use to actually get your data mapped to the standards. So for example, you would start with the source data, then you would get some report about what type of data is in your data source and how can you then best map that to the OMOP CDM using this tool. It's called Rabbit in a Hat, and Usagi is the one that helps you with the semantic mappings. And then what you see here below is more the infrastructure, because you want to make sure that you can, for example, use the analysis packages that have been developed by the OHDSI community. So what you see here, these are the logos for Atlas, which is a web-based tool that allows you to create cohorts and to run analysis packages. And the Hades suite is already a set of R packages that can be used for the analysis. And so during this project, what we saw is there's a lot of hospitals, resources, registries. There was a lot of interest to be part of this big network. So as you can see here, during the project, we had like 560 applicants. Not all of them could get the funding, of course, but in the course of this project, we mapped 187 of those resources to the OMOP CDM all over Europe. So as you can see here, there's a little bit of a distribution for the different countries. And so this was as part of the Eden project, but as I said, this project has ended in October last year, but now the Eden Foundation is taking over. So that's a not-for-profit organization that still allows for large-scale analysis using this network of real-world data sources in the OMOP CDM. And if you want to know more about this or dig deeper into the sources, you can go to the Eden portal. So I think the link is also on the slides that we will share later. There you can request an account, and there you can really use those two aspects, for example, the database catalog and the network dashboards. And in the database catalog, you really get all the metadata about the different data sources. So for example, what's the description of the data? What type of data is there? What's the geographic coverage? How many people are included? But also about, for example, the governance and the ethical approval processes, so that you know for what you can use the data. And then on the other hand, you have the network dashboards that really allows you to see high level what data you have in the different sources. So for example, this is an overview where you see this, as I said, just the overview. You see per country how many patients we have. We have 30 countries represented. We have 208 data sources and more than 460 million patients that are represented at the moment. And what I don't show here, but what you also can, dig a little bit deeper, is then really the demographics, for example. So you just see the distribution of the genders or the ages or the observation periods, how much data you have per patient in which of the domains, for example. And also you can use the concept browser to really see for a certain concept ID for a certain disease, for example. You can just filter out how many patients you have in the different sources, so different hospitals, for example. So that really helps you if you want to do a feasibility assessment for large network studies. But that is Eden and the Eden Foundation. And this builds actually the foundation, I would say, for the other two projects that I would like to go into now. So there we have IHI-GREG, which is also, a public-private partnership that just started in May this year. And here we're actually trying to figure out how we can provide guidance and tools to really create and test and share the practical implementation of those of the creation of real-world evidence. So how can we use the real-world data that's already out there, standardized and high quality, to actually improve the decision-making for the regulatory bodies and HTA bodies, for example. And then we not only focusing on the medicines, because this is already mapped quite nicely, but we also want to extend the data mapping to include devices, for example, and drug device combinations, which also Berber showed already. This is a little bit not more tricky, but I think that's a really newer to include also those devices and the data we get from there. And so, as I said, just started in May, and there's a nice little circle that we go around, starting from the evaluation. So we want to look at what are the existing real-world evidence guidance or recommendations. And then we're going to look at a couple of use cases from regulatory and HTA bodies, which were successful, which were not successful for the decision making. And then we want to co-create new guidance and new tooling to actually improve that together with the public and the private partners. We would elucidate then with with the people that are the stakeholders to really understand, okay, what's important here, and then start to create new toolings and new guidance and do the pilot testing based on, for example, that network that we saw in Eden, because the Eden Foundation is part of GREC. So we can actually use those guidelines and those tools that we develop here on this huge network all over Europe with those 200 data sources. And then, of course, after the pilot testing, we would consult again with some of the advisory boards and see whether that makes sense. We would do the training and dissemination and then just go to another use case, because there's obviously a lot of use cases that people are interested in when it comes to drugs, devices, and drug-device combinations. And so the last project that I would like to also present to you is the Darwin EU initiative, which is started from the European Medicines Agency. As you know, this is important, or this agency is important to facilitate the development and access to medicines in Europe. And they also evaluate all the applications for the marketing authorization for new drugs to come to the market. And there's also a lot of safety monitoring that they have to do with all the medicines across their lifetime. And so a couple of years ago, the big data steering group from the EMA and all the HMAs decided maybe it would be good to not only look at clinical trial data, which is of course important, but we should also use the opportunity to use real world data. So this is the routinely collected data that Berber explained and that you have heard also. So that's the hospital data, registry data, GP data, mHealth data, and the claims. And then mHealth is a little bit mobile, for example, where the social media falls under, but also some of the devices. And so when they started out with Darwin, they had the idea that by 2025, which is now, obviously, we should use real-world evidence also to make regulatory, or to help make regulatory decision-making. And in the next couple of slides, I want to show you where we are with that. So first of all, the Darwin EU initiative is set up in a way that the EMA and all the different committees there can ask questions to the coordination center, which is Erasmus MC in Rotterdam. And they have a couple of subcontractors that help them to run actually the studies over the data partners that we saw. And also here there's quite a number of them that are already based on the Eden network that we saw before. These are the partners, so Erasmus MC, as I said, and there's a couple of subcontractors for different skills. So for example, for the project management and more epidemiology knowledge and expertise, but also data quality and vocabularies in the context of OMOP and OHDSI. And so this is actually what the whole Darwin group provides to the EMA. So there's, of course, a lot of knowledge and a lot of expertise about how to run studies. There's the catalog of all the data sources that we know. There's the maintaining and extension of the data network that we have for the federated analysis. And also Darwin really conducts those scientific studies. And besides, we're also training and helping with the governance for the sites, but also for the EMA. And so this is just an overview of the type of studies that we're running. So there's a little bit of a hierarchy included here. You start with the routinely repeated analysis, which is not super difficult. Then there are off-the-shelf studies, complex studies, and very complex studies. And then, of course, the routinely repeated analysis are the ones that don't take that much time, and the complex ones are the ones that take a lot of time. And here's a little bit of an overview how many studies that actually are per year. So we set up the whole Darwin network from 2021, I think. So there's not so many real-world data studies involved. Now we're in the scale-up phase, and from now on, the idea is to run up to 100 to 120 studies using real-world data to create real-world evidence every year. And so coming back to the network, so this is also, of course, growing because we're adapting and adding 10 new data partners to the Darwin network each year. So at the end of this year, which ends in February, there will be 40 data partners that are being included in the network. And then, so the last point that I want to focus on, I think I have to speed up a little bit, is about the data quality insurance, because as you can imagine, for those regulatory studies, you want to assure really high data quality. And there's, fortunately, there's also a couple of tools that have been provided or developed and provided by the OHDSI community. They are also on the screen here. So the data quality dashboard, Achilles, and the CDM onboarding. And the process that's being followed now is, that we review the output of those quality assessment packages. Then we talk to the data partners to help them improve their mappings and improve the data quality, and then to come back with the improved data quality. And so I would like to shortly go into how the data quality dashboard works. So that's based on the Khan framework, where we look at verification and validation of data, where the verification really focuses on the internal benchmarks and the internal expectations, where validation focuses more on the external benchmarks. And there's three categories that we're looking at. So conformance, really, does it fit into the expectations that we have looking at the format of the data? Completeness is there any missing data? And plausibility focuses more on, for example, that we don't see any procedures after death, for example, or that we don't expect prostate cancer diagnosis in a female, for example. So this also helps you to assess the data quality. These are some examples where I don't go into now, because we also want to have some time for some questions, but we will share the slides so you can have a look at those examples. And then for the data quality dashboard, you also have a nice R-Shiny application where you really can see all the passes and the fails for those different categories, so the verification and the validation, together with the plausibility, conformance, and completeness. And you also get quite a nice report where you can see what actually failed, so that you can discuss that with the data partners. So either it could be that they know of this issue and they can explain it, or it's really something that went wrong in the mapping. But this really helps you to ensure, for example, the lineage and the data quality. And the other tool that's being used is the CDM onboarding. It's just a package that gives you a little bit of information about what's in your data. So for example, what are the package versions that are being used? What's the CDM version and the vocabulary versions? And then you can see whether the core tables are filled with the data you would expect. And also you see an overview of the records in the different tables that you would need. So that gives you a nice overview. It contains information about the date range, so you can see whether that all makes sense, that the start date and the end date, for example, are not somewhere in the future or super far away in the past. And then also for the vocabulary mappings, you see how much of the codes have been actually mapped and which haven't. So that also gives you a good indication what's in your data. And then since we especially do that for Darwin, where of course the drug mappings are really important, you can also see what are the levels of drug information that has been mapped. So is it like on an ingredient level, or is it just a clinical drug, or is it a certain brand name? And this is also, if you want to work together in collaboration in a network, that is of course where you want to agree on. And with that, I would just share some of the further readings that you can take a look at. So as Berber also showed you already, there's a lot of info sheets and reports available from the EMA where you can read up on this. And there's also the HMA EMA catalogs where you can find all the real-world data sources and also all the real-world studies that have been executed in the past. And I think this is also a really nice resource for the learning. And I think now it's back to Berber for the key takeaways, or we might need to take some questions, I think, since we don't have much time.
I would say the questions.
Indeed. Okay. All right. Then I'm going to jump in with the questions. Julia, thank you so much for this presentation. I'm going to quickly jump into the question for Berber by Stefano. He asks, I think this was reference to the slides that you showed after mapping with SDTM and OMOP. He asked if you can say something about the mentioned data standards and domains, especially in terms of mapping capabilities.
Yeah, I'm not completely sure what was asked, but if you look at mapping from OMOP to SDTM, then OMOP has they have the less different tables if you look at the clinical data. So you have your observations, you have your measurements, you have your conditions, etc. Well, in CDISC you have many different domains, as I already explained, for medical history and adverse events. So yeah, the mapping is doable and also the coding, but it's it's better to do as not all the data at the start, but select the data that you need and do the mapping. Not sure what's mentioned by the mapping capabilities, but I think there are mappings available already from OMOP to SDTM, so I think there is also something available there already.
Yeah, I think he's kind of expanding on his question as well. So in the meantime, then I'm going to skip on to a question. I think this is for Julia, where Anna asks, which data quality aspects are most important when collaborating in a real-world data network that leverages OMOP?
Yeah, good question, I would say. I think it really depends on the purpose. So what do you want to analyze, right? So of course there are some mandatory fields that really they have to be there. But for example, when it comes to the drug mappings, you would say for something like Darwin, where you really focus on drugs, you want to have all the items mapped to the really lowest level of specificity, for example. While if you look at something else, this might not be important. So I think it's really important to see, okay, what do you want to focus on and what do you want to analyze?
Okay. All right, thank you. And then I have one more question, and this is for Berber. What improvements do you recommend for the lineage of real-world data?
Yeah, actually two things. I already got this question. So one is already think beforehand what the information is that you need for your data lineage and set up your structures for that. So that's one. And the other one is I forgot. I will come back to that. So the structure is important, and also the standards, also the mapping information, standardizing the mapping information. What we see now is that a lot of information around the mapping is just written in words, and then it's still subject to interpretation. And what I want, that's one of my goals for the industry, is also standardize that. And that's what we're also doing with that tooling, that you see, okay, we can also standardize the recode information, the mapping information, and also the coding behind it, so that there's no interpretation possible, and there's no subjective interpretation anymore.
Alrighty, thank you. And no, I think these are all the questions that we had from our audience. Well, I had one more question that was for Julia, where I want to ask, if we were to start with data quality assessment on my OMOP database, what would be my best place to start? I mean, you briefly touched upon it towards the end of your presentation, of course, but I thought...
Yeah, I think I would start with looking at the tools that are available, right? So I think because they are open source tools, so you can just get started with them. But there's some sort of a learning curve involved. So I would also start with looking at the resources around them and just try them out and see what's possible, and also reach out to the community, because there's a lot of experts that are really helpful. So if you have questions, I think there's also a lot of people that can get you started there.
All right, perfect. Thank you. And I think with this question, we are also nearing the end of our hour of webinar. So I'm going to take this opportunity to say, if you have any more questions, our people in the audience, please do reach out to us and we will connect you with the right experts to answer them. Thank you, Julia, and thank you, Berber, for this wonderful, wonderful webinar session. As always, the recordings for these sessions and the slides will be available to the participants who have registered for the webinars. So that will be available to you. And since we are coming towards the end of the year as well, in a few weeks we have our winter break. So on behalf of the Hive, and we would like to wish you all a lovely winter break as well. And if you are interested in webinar series such as with OMOP, then please follow us on our LinkedIn pages, because we will also be starting a new webinar series starting January. So please keep a lookout for the LinkedIn pages as well. I wish you all a wonderful continuation of your day. Thank you, and see you next time.
Thank you.
Thank you.